Second talk of this session will be given by Anupam Gupta from IIT Hyderabad. He will be talking on modeling embryo elongation and somitogenesis. Over to you, Anupam.
Thank you. Thank you very much for the introduction. So before I forget, let me thank Ranjini and Mahesh for organizing this nice workshop and inviting me here to talk. And also, I would like to thank my funding agencies because I usually forget. So IIT Hyderabad, SERB, NSM, and all. Okay, so what I'm going to do, I will talk about how to model like early embryo development and then how the somite formation happens in vertebrates. So it is highly driven by the model is driven by experimental observations. So closely, I will try to show experimental results and how we can tweak our model to observe those results. So that's the aim of this whole talk today. So before I start, I'm not sure how many of you are familiar with embryo development. So I would like to show a video. Yes. Okay. So this is a chick embryo, as you can see, and this is how the embryo development. This is how the full embryo development looks like. In the beginning, it starts from a spherical structure. Then, as a physicist, we say it breaks the symmetry, starts to elongate. Then there is a left-right symmetry, and then it breaks that symmetry, and this is how the overall development looks like. So, as I said, what I will be interested in, I will be interested in only the early part of embryo development, which is related to this part. Like once it breaks the spherical symmetry and it starts to elongate, here you can see. So this part is embryo elongation, and there is a steady state, right? So the embryo elongates with a constant rate of elongation. So that's what I would try to reproduce using my models. And the second part is there is formation of these somites. You can see the small dotted structures on both left and right side. Okay, so these are known as somites. So these two things I would try to explain today, and I would try to develop a minimal model for that. So okay, so to give you more insight, so how the structure looks like. So basically, if you take this portion, you will see that this is this end is known as tail. We all have it. It doesn't means it's there. These are somites. There is a left-right symmetry you can see. In the middle there is a neural tube, and these are lateral plates. So when these tissue formation, early stages of somites, these plates are highly rigid. Okay, these neural plates, lateral plates, so they act as confinement. So all the dynamics happens in between these plates. And also at this stage, the tissue is more fluidic in nature. This is more in mixed phase. With time, the motility of these cells. So I will explain these things in detail. So in the beginning, it is in mixed phase, but after that, there is a phase separation starts to happen, and that leads to these structured formation, and we call them somite. Okay. So basically, so you can see that there is a left-right symmetry. So what one can do, one can actually focus only on one half of it and try to explain it. Okay. Okay. So before I jump to the model and show any more movies, I would like to address the open questions. So it is still not of course we have some understanding, but still we do not understand very well how the means what is it which controls the embryo development. For example, body elongation, its initiation. So there have been many experiments where they start from a spherical structure of these early embryos, and then they try to genetically or using some chemicals, some proteins, they try to bring in activity and see if that leads to symmetry breaking. But there have not been many successful experiments. So how the symmetry breaks and it starts to elongate. Uh, so that is the initiation process. How does it terminate once the elongation starts to happen? And then what is it which leads, why there is a steady state? How can one reach that? So these are the questions related to body elongation, which is this one, how it elongates. And then second question is somitogenesis, these small structure which appear like this is a skeleton of snake. So these structures are known as somites. So these are open questions. Other than that, is this mechanism same for different animals with vertebrae, like for us, for snake, mice, chick, and zebrafish or any vertebrate animals. So as you already know that we have close to 33 somites, whereas if you think about snakes, they have close to 300 to 400 somites, right? So the difference is huge. So can we use the same mechanism to explain all these somite formation? Okay, so that is another question. Uh, Another open question is what is it which actually drives? Is it gene expression or physical, or is it a physical process? So if you talk to most of the biologists, not biophysics or development biophysics people, they say that everything is driven by gene expression. Whatever mechanical properties come, it comes through as a result of these gene expression. Whereas if you talk to any biophysics person, they will say that it is a combination of it. It cannot be alone. If there is a mechanics, if cell has a size, it has some contraction, some tension. So of course that should lead to some mechanical forces, and that should lead to some it should be able to drive the system. So what here we'll try to do, we'll try to say that okay, once the cell type have been differentiated, once the gene expression has been given to the cell, can physics only drive the elongation? Can physics alone drive the somitogenesis? So this is what we'll try to do today. Okay. So, so now, let me show you. So now we are heading towards the modeling part. So this is a movie which is not playing. Okay. So this is an actual experiment on chick embryo. The green color thing you can, so this is the neural tube. This is the source of new cells which are coming into the this mesoderm. We call this portion mesoderm where tissue is. So new cells are coming from this point. These are highly motile in nature. And so in that way, you can say, as a physicist, if I want to look at it, so there is addition of mass, and these are motile, so there is activity. So together they are trying to elongate the body. Okay, and at the end you can see that there is somite formation. So in experiments, one can do single cell tracking for these newly incoming cells, and one can see. I don't know the actual tagging. Means it is some G, some fluorescent. I don't know. I'm really not good at green fluorescent protein. GFP. Those are stem cells. So it starts from stem cells, but then they start to differentiate. Embryonic development. This is that. Okay, is that so? These are not epithelial. So once somite starts to form, then there is the epithelialization. So no, these are the rib cage or rib. Yeah, yeah, that's what. So somites are actually rib cage. Just to get you clarity. Okay, at the end of the day, they become our rib cage. So, so yeah. So if you do the single cell tracking of these new incoming cells, so this is so also we call this end as anterior end, this end as posterior end. So we'll be referring to it a lot. Okay. So if you focus at how the cell moves at this end, close to posterior, and this is the single cell tracking for this end, you can see that it moves a lot. Whereas if you look at the single cell tracking in this region, which is the anterior region, you can see that there is not much motion. Okay, so this is one way to determine how active these cells are, how motile these cells are. Okay, we have already seen here that new cells are coming into picture. So can we use using this information? We already know that there is a gradient in activity, motility. Okay, new cells are coming into picture. Also, we know that there is confinement, that there is a neural plate, there are lateral plates. So can we use this information and build our model and see whether our model works or not? Okay, so this is what we'll try to do. Yes, so this is a sketch just to give clarity. So this is how, at the end of the day, new cells would come from here. We call this area as pre-somatic mesoderm. So pre-somatic because they are going to be somites. So before becoming somite, we call them. So that's why the name comes. Also, in terms of genetic expression and protein level expression, we know that it has been experimentally observed that when you go from posterior end to anterior end, there is a decrease in FGF protein level, and that is directly related to the motility of the cells. Okay, so one can put it by hand. We know that it is there. Of course, new cells are coming in. We also know that rate at which new cells come in. Okay, and so basic. So what I'm trying to what so for this problem the question is this one forward backward. Okay, thank you.
These cells which are previous slide in the movie that was-
Right.
So the cells are originating at one place.
One place, yes.
Okay, they are getting some sort of frozen structures are coming in after they make somites, right?
Yes, yes.
They
Free, they actually free. So basically, in the beginning, they're highly motile. So even though they have adhesion, but they cannot form clusters. But once the motility goes down, adhesion starts to dominate, and that leads to clustered structure.
But these clusters are also very much
Very regular, right?
regular.
Yes, yes. So there is an explanation. So we'll come. I will explain that why it is regular. So there is a model for that, and it is also experimentally verified that there is an oscillation in genetic. So you know that there are so many proteins which come into picture. So one kind of protein which are different from for all these animals, but one protein always oscillates during early stages of embryo development. So those oscillations and the where the somite starts to form, so that wavefront is moving forward from interior to posterior direction. So together they give these regular structures. Okay. What's the? Okay. Thank you. So backward also works, right? Okay, I will point to this direction. Okay, so the question is only based on these information that cells are coming in. And also one more important thing which one should note that this motility is random motility. It's not like a directed motility. Okay, and this has been observed, means reported extensively that these motilities are random. Means they don't have any directionality. Okay, so can random motility and of course addition of mass and confinement, can all these three together, if we combine them, can it lead to embryo elongation or not? If there was, so I will, so if there are no confinement, it will grow isotropically, but confinement is the one which leads to a directed elongation. Okay,
So
yes.
here,
Yes.
yeah. So I had a question. It's like a cell is coming from somewhere, or they are growing also along with that, right?
There, what happened? This is pointer, right? Yes. So cells means they get generated here. So
So,
throughout the process, we are not considering any cell division. Cells are not getting proliferated in this region. Cells are mainly coming from this point. So they are coming from outside. You can
so actually that happens. That is you are assuming in a model.
no, it is a fact.
It won't divide. Okay.
It is a fact during this process.
Okay.
Means this one dominates. Okay, this one dominates. So there is not much cell differentiation over the process. As division.
Okay.
Yeah. Yes, it flows back. Means when the cells enter, so basically, when the cell enter and due to activity, it starts to move this boundary. So tailbud moves forward. So it will apply. So active pressure and addition of mass together leads to that. So I will show another experimental observation.
Local site in which there will be high density of cells. So it's sort of like, in physics language, chemical potential, strong chemical potential gradient
So
generated because you are injecting new particles.
Right. So, but throughout, so if you do an averaging along this direction, it remains constant. Cell density remains. Total number of cell density remains constant. Number density, I mean, number density remains constant.
Local density,
Uh,
because it grows.
yes, yes.
Yeah.
It's like there is a wave of cell, like there is a traveling wave of those embryo cells.
No. Means these are just being injected to the cells. They just enter your system. This is the domain, right? In which they just enter. They get generated and then they enter your domain. Okay, you can see in this video.
Okay, thank you.
I should have put it in the loop, but so it is. So these cells are already there in the neural tube, and it gets generated and it comes into the domain where we are interested in. Okay.
Looking at this movie, it looks like the cells are moving in a very directed fashion, right? From the tail to
No, so they remain more or less there. They just randomly move around. So of course, few of them can go in, but it's not like the cells go in. It's not like that. Okay, they just stay there. The boundary is moving. So basically, it's like this is the thing, right? You are fluctuating, putting pressure, and it will keep on moving. Then new cells come, and again you apply pressure. So it will keep on moving. So new highly active cells come. They will apply pressure. It will move. Then again, new cells will occupy this space. So in that way, you push, create space. New cells will come. Again, you push. New cells will keep on coming. So that's how it works. Okay, and also its motility goes down with time. So with age, its activity goes down, and that leads to at the end condensation. But that condensation is supported by another protein level oscillations. Okay, and that leads to this rhythmic um, pattern, means nicely spaced structures. It is, I think, 0.3 micron or per second. I need to check. There is the number. I don't remember exactly, but few microns. You mean the right, right. Uh, I think both of them are more or less of the same time scale. So if suppose if diffusion is much small, like this diffusion, then it will not be able to apply pressure. So diffusion time scale should definitely be slower, longer than elongation speed. So elongation is slower than diffusion time scale. Okay, if diffusion is slow, then it will apply pressure, but it will not be enough to move it. It is confined. Means there is a neural plate here in between, and then there are lateral plates on the side. There is confinement. So it is not hard confinement, soft confinement, but these cells cannot go out of it. Okay. Fine. Any more questions? So this is how it looks, and then I will start the modeling. So all the assumptions are based on the observed facts. Okay. Okay. Fine. So how do we build? So as I said, I will. One can look at. One can take the symmetric half of these embryo and then try to explain it. So what I will do, I will take this part. This is how it is going to look. As a physicist, we can always make things either circle or rectangle. So I have made it rectangle. And this is the tail bud. And I assume that new cells enter from here. The condition for new cells to enter the this pre-somatic mesoderm, PSM, which we call often. So there should be enough space for these cells to enter. If there is not enough space, it cannot enter. Okay, and how will we generate enough space? If you displace your tail bud, only then, only in that condition, there could be new cells can enter. Okay, then other than that, these cells have a lifetime. The cell motility goes down as they age. This is a zoomed in image. This is how it overall looks like. Let me write down the equation. So we use overdamped Langevin equation here to, so the Okay, let me put everything together and then I will explain. That will be easier. So here I consider that there are no inertial effects, so I can go for overdamped Langevin equation. This is the potential. This is the noise tuning which I control the activity. There is this M parameter which sets the activity. This M parameter decays with a time scale. I say that if a cell is older than a certain time, then it will decay with some exponential rate with this probability, and this is the motility time scale. So this tau sets the how slow or fast motility is decaying in time, and since things are moving, so it gets reflected in space as well. Okay. Then for the potential, I'm considering the particles to be soft and there is simple harmonic potential. So when they are in contact, only then they will try to repel each other. Otherwise, there is no interaction. Okay. And okay, so interaction I have defined, the noise term I have defined. Now, in terms of time scale, which you were asking, so one can define a time scale which will, so basically use from this motility, we can get diffusivity. If you do m by gamma is the friction coefficient, that gives us a time scale. So basically the decay times, so the motility time scale which is associated with the decay of activity of these cells, this should be much longer than the diffusion means this diffusion time scale. Okay, and that together leads to embryo elongation. So what I'm trying to say that suppose if a new cell enters here and if it loses its motility quickly, then it will not be able to apply enough pressure on tail bud. So it has to sustain its motility for some time, so that it can apply a pressure on the tail bud and then it should be able to move. Okay. And then using some dimensional analysis, one can actually get a speed of elongation, and that is going to be related to the local density close to the tail bud multiplied by this d, and then a is the size of particles, cell size in this case. Okay. Fine. So if you solve this equation, again, another thing, to take into confinement what I'm doing, I'm using a periodic boundary condition in this direction, in the lateral direction. So neural tube and lateral walls, I say that there is a periodic boundary condition. In that way, it sets the confinement. In the interior region, I'm saying that once there are somites, as they means there is an arrest, these guys cannot move, so I can use a rigid wall boundary condition. So I'm using a rigid wall boundary condition, and tail buds are modeled as a spring of beads, which with some spring coefficient, and they're free to move with some friction. Okay. That changes with time. So at the beginning, when a new cell enters, it has a motility m, but this motility decays with time exponentially. Yes, this form. It goes to means very small value. It goes to very small value. Okay. So basically, in the experiments also, one can do the particle tracking. From that, you can look at the mean square displacement, and you can look at how the diffusivity decays in time and drift velocity. So from that, you know that you can set a length scale, right? And that length scale can be converted to a time scale because there is elongation velocity. So that time scale you can use here in our model.
So can I ask a question?
Yes.
So is the driving force in your model is the collision between these particles?
And the tail bud.
So particles which are there in the tail bud, right? The collision between them, because that U is coming from that spring action, right?
So this I represents the index of the cell. I have not written the equation for how the particles interact with the beads of the tail bud. So basically, they collide, and there is just purely repulsive interaction. So whenever they hit,
that is what is driving the movement.
right.
Okay, and then there is no net average force acting on the entire system, right?
No, on the spring there is no other force acting. So this is again an emergent feature.
Other than that random force.
Yeah, of course, there is random force. So these cells are active. Okay. Yeah. So basically, these are like attractive, slightly attractive in nature. these cells. So I'm not putting it in the model because here I'm only interested in how the elongation looks like.
No, what I'm asking is that this UI is zero.
Right. I agree. I agree. Yes.
And this noise will go to zero for the whole.
Right.
Then they will just be
Yes. Yes.
some kind of material which doesn't move.
Right. Right.
Only the new cells will move.
Yes. Precisely.
New cells are at the tip of
At the tip of this point. Yes. Precisely. But this was a very basic model which we started with. But eventually, what I will show that you can bring in attraction as well, and that leads to condensation. So this was the first thing which we tried, and then I will slowly show how it evolves. Okay, now I will. Let's see how the simulation looks like. So basically, this is the tail bud which is moving. These are the highly motile cells. You will see over the time here the motility will go down. Okay, this is the anterior posterior direction. This is the anterior end. This is the posterior end, and you can see that there is a steady state and it moves. So in the beginning, it takes some time to pick a steady velocity, but then eventually it reaches a steady state velocity. And we have also checked that if you control your motility decay time scale, you can control means you can actually terminate this elongation. So if you say that you have a situation where your motility time scale remains constant for some time, and then it comes down. So this is my tau motility time scale. So if you said that, and I'm talking about this tau only, means tau is fixed for all the cells, but I'm saying that if we make it time dependent, in that way you can actually stop the elongation. And that's what happens in the experiments also. Retinoic acid comes from the posterior end, and it kills all the FGF protein. So basically in that way it kills the motility of the cells in this area. Okay, so that can. No, there should not be any density gradient, right? Because cells are there. So there will be, so yeah. Right. No, why do you, mean cells are there, right? Cells are there. So new cells come, it moves, then again new cells come. So there is no way that it will keep on growing here. Then it will just explode, right? If you think. And we are limited by this space. Space is a constraint. You cannot go beyond certain. So that is, it is an experimentally observed quantity and varies on different species. Here, what we have done, so I will show in this plot that, so that's the beauty of simulations. You can play with all the parameters, right? So here we are changing the number density in the neighborhood of tail bud. So basically, I'm saying that I will maintain a maximum density packing fraction here. Rho m is the packing fraction, the number of particles in a given area, and d again comes from the motility m and gamma, and what here I'm plotting the position of the tail bud. So this is how this is moving. So it starts from some certain position, and then it is linearly changing with time. So it sets a steady state elongation velocity, and here I'm plotting a normalized plot. So basically you can normalize your time with rho, m, and d, and that's why you can collapse all the curves. So if suppose your rho, if you change your rho m, and if you look at unnormalized plot, what you will see that with more number of active particles, your tail bud will move faster. In the same way, if your d is higher, which is motility is higher, again your tail bud will move faster. But when you rescale your time with this, you can collapse everything. Okay?
So here,
Yes.
I have a question. So what I'm trying to say, at the front, cells are growing, and because of that, there is a moment. It's a moving forward because of that. And after some time, they are becoming non-motile, but front cell again growing and moving.
Yes. So what I'm saying that there is a constant influx of cells, okay, with a given
Local, right? Local influx.
local at the tail bud at this point.
And that is coming because of cell division only, right?
Yes, somewhere cell division is happening, but not in this domain. It is coming from outside, from the neural tube.
So my question is that only, like in left side, cell is not moving,
Right.
They're non-motile. then how cell are coming?
Well,
It makes
coming
sense
from this
only at the
end.
front if they are growing.
So,
No?
okay, if you want to you can also say that these cells are dividing like crazy.
Yes.
Every 10 minutes they are dividing, which is not possible, which we know that biologically it is not possible. Okay, so cells are coming from outside. Okay, outside means the neural tube, everything you remember if the video
Uh-huh,
is
yes.
coming from there. So that's why I don't want to say that these guys
So
are dividing.
tube is also growing with time because embryo size is growing, right?
Tube is moving, yeah, everything is moving forward.
And
as
growth is
everywhere.
happening inside tube only, cell growth.
Cell growth, yes, cell generation is happening inside. New cells are proliferating in the neural.
Okay.
Okay.
Thank you.
Yes, please. So these are highly means overdamped setup. So no instability will grow. So whatever it will get dissipated. Where is so again I'm saying that there is no density difference if you go from interior to posterior direction. Okay, I'm saying that there is a constant influx of cells. Things are moving forward. So what I'm saying, you move this guy and you keep adding things. Okay, so you can maintain a constant density in this direction. You can do that, right? Fine. So again, I will repeat, there is no number density is not changing if you go from interior to posterior direction. Okay, fine. So, okay, experimental observation which Saroj was asking. So basically, what one can do, one can actually track these cells in experiments, and from that one can look at mean squared displacement, and from that one can extract diffusivity and velocity. Um, so, what I wanted to do, yeah. So since activity is going down, corresponding diffusivity also goes down. This is the posterior end, this is the anterior end. Okay, the same is true for the drift velocity. So the drift velocity is large at posterior end, and it goes down as you go towards the anterior end. And again, to tell you the number, Sanjeev, it is order of 0.03 microns per second. Yeah. Okay. Yes, so this is an experimental observation. Now what we'll do, we'll try to look at our data and see whether we can capture the similar things or not. Okay. So, so again, we can do the same thing. So in the simulation, again, we know exactly where the particles are. We know the cell tracking. And from that, we can characterize what is the diffusivity, what is the drift velocity. I'm not giving you the details how the mean square displacement. I think all of you know because we have learned a lot of glassy system talks, so we know that. So this is the relation which I'm using. And depending on different motility time scale, you will have different decay profile. So this motility time scale sets what is the length of pre-somatic mesoderm. So it also sets like this time scales for how much time, but also in space it sets a length scale. When if you use your elongation speed and this motility decay time, that gives you a length scale. So that sets the pre-somatic mesoderm size also. Okay. So you can see for different times, there are different length scale which are coming into picture. Similarly for drift velocity. But if you collapse it using the length scale which I was talking about, which comes from diffusion, from the velocity, elongation velocity and the time scale, then you can collapse all of these. Okay. Now, so we can see that there is a similar trend. At the end, I will also compare the experimental and numerical results. So this is about the microscopic model. What one can do, one can also use a continuum approach and write down the mass balance and stress balance equation. So how will you do that? So now in this case, we are talking, so as all of us have been saying, cell density gradient. So there will be a cell density gradient, but that will be in terms of motile and non-motile cells. So total number of cells density is going to be fixed. But if you think about how many cells are going to be motile and how many cells are going to be non-motile or in terms of effective motility, so you can see that there is a decay. Okay. So this row is representing the motile cell density. These guys are getting advected by the drift velocity, which is inside the PSM. And then, of course, they are diffusive. And there is a degradation which is related to motility decay time. So that you can, so this sets how fast or slow the density of motile cells are decaying. Okay, so this will be our mass balance equation for the motile cells. We are not interested in non-motile cells. When we go for the complete model, at that time we'll worry about how the non-motile cell density, how the motile cell density, and also there is extracellular matrix, how that dynamics is changing. Fine. One can also write the force balance equation. We are in the non-inertial limit, so we'll directly write how the means you can write forces from this pressure. That will be basically active pressure and the viscosity within the PSM, because the cells can generate an internal viscosity. And then these guys are moving on a surface, right? So there will be wall friction. So that's how it loses its energy. Okay, so this is going to be our equation for force balance. Now, if you solve it, so again, I'm not talking about somites now. I will be talking about somites later. Another thing. So there is a in this equation, if you think about boundary conditions, so this guy is moving forward. The posterior end is moving forward. So there is a moving boundary condition. You at the end of the day, when you solve your PDs or even microscopic model, you should have a steady state, and we'll see that we achieve that. And if you look at the anterior end, we know that there should the motile density, the density of motile cells should go to 0. In the same way, drift velocity should also go to 0. So that is the boundary condition here. Uh, yeah, that's the boundary condition. Yes, yes. So basically, R is the rate of injection of cells. And we are saying, as in the earlier case, also in the neighborhood of the tail bud, or in this case, the boundary condition should be such that it should maintain a constant density at the tail bud. Okay, so rho 0 is that. Rho S is the boundary density at the interface. S is representing the position of the boundary. Okay, so rho 0 minus rho S, if you multiply by R, that will give you the left hand side of the mass balance equation. And this will be balanced by the right hand side of the mass balance equation, which comes from the diffusion and the advection. So advection at the boundary, rho at the boundary S, and S dot how fast this tail bud is moving, and then there is minus d rho x. So this will be one boundary condition for the mass balance, and then similarly there will be a boundary condition for the momentum balance. So basically what we are saying that the active pressure and the viscous dissipation term should be balanced by the resisting force of the tail bud. Okay, so if we solve this equation, and again rho at the anterior end and v at the interior end should go to 0. So if you solve this equation, this is what you will get. So this is how the velocity drift velocity profile looks like. This is from the simulation. This line, this light green dots are from the experiments. Similarly, for the diffusivity, these lines are from the simulation. The dots or circles are from the experiment. This is the elongation speed at the tail bud. You can see in the beginning, the elongation speed grows and then it reaches a steady state. Okay, and similarly for diffusivity, this is how it looks like. So we again. We had to do a lot of, so we had to scan the complete phase space to find the correct parameter. Otherwise, it was impossible to fit the experiments and theory data. So that I have not described here. So now our minimal model, so the take-home message is that our minimum model is able to reproduce the embryo elongation, random cell motility without any directionality with confinement is good enough to give us elongation. And this will be, I have 20 more minutes, right? We started 5 minutes late, right? So, okay. So in the next one, I will actually show that how it is important from experimental data. And then I will also extend this model and I will show how robust this model is. Okay. So again, just to summarize, this is the result of two effects. Addition of motile cells. Motile cells have random motility and confinement. So if you combine these two, you will get you are able to get this elongation. So so till now, even in the experiments, what results I have shown, I have mainly shown the results from the actual embryo, growing embryo, which is attached to the egg or zebrafish, so all those things. But now what I'm going to do, I'm going to talk about pre-somatic mesoderm explant. So basically, this is our embryo. This is a sketch. You can cut it, take it out, and do the mechanical measurements such as mechanical properties. Okay, and then what we have done, whatever experimental observations our experimental counterparts have done, we have used the same model and then we have tried to show that how good or bad our model is, even though our model is minimal. In biology, there are so many things come into picture, as I have been saying that always there are 5-6 proteins active to regulate things, which we are not putting any of those information in our model, okay? So if you cut this explant and you just look at how the relaxation and everything, so what you will see that it starts to relax, and then there is this kind of formation, and if you wait long enough, then it relaxes to a state, and there is a pear-shaped formation. Okay, so that is an experimental observation. I will we'll see why we observe such things. Why there is this. So why doesn't it grow uniformly? Of course, we can guess that activity levels are different. There is a gradient in motility. So of course, that is the one which is responsible for this. So if we want to model it, how do we do that? So till now, what we were saying that we were using a rigid boundary condition at the anterior end, right? Rigid wall. In the lateral direction, we were saying that there is a periodic boundary condition. And at the posterior end, we were saying that we are using a spring to model this tail bud. But now what we are saying that since it is free, there is no confinement. So we'll uh, confine everything with a spring with uniform spring constant. We are not changing the property of the spring coefficient or any such property of this boundary where this explant is attached. And basically, this is a signature of that there is epithelialization is happening. Okay, and so which we are putting by hand in our model, which in experiments happen. Once you take the explant out, you can see that even though they are mesenchymal, but they will start to epithelialize. Okay. So again, there is no change in model. If you go from posterior to anterior end, there is decay in motility. The spring size I have considered to be same as the cell size, and yeah, and rest of the dynamics is exactly same. So now, if you let's try to do a comparison between experiments and simulations. So as I was saying, that if you look at uh, the PSM explant in the experiments. So you will see that first it starts to relax, it will start to contract. Lengthwise, its length will reduce. Whereas in terms of width, it's towards the posterior end, its width will start to increase. And the simple reason is being that in the posterior end there are more motile cells, and that they can apply active pressure. Okay, if we do the same thing with our simulation, just by bringing in um, uh, just by bringing in the motility gradient, we observe something similar. So you again start from a PSM of this size. The color bar shows the motility value. So red means highly motile, blue means close to zero. Okay. So we can see that it is not means it is approaching the same thing what we have seen in the experiments. One can measure such as the means how the length of the PSM is changing over time in experiments, and also how the width. So by width they look at the maximum width. Okay, and that's what they have seen. We have seen something similar. It is not exactly same, but something similar. So we see that width wise there is enough growth, but in terms of length, it does not contract in the same way. Yes, please.
If I understand the model correctly, you have these four sides.
Right.
You have the beads which are connected with the springs on the four sides, and you have cells
Right.
with different motility
Right.
and different sides.
Right.
And what you're saying is this bulging or the pear shape is just coming because of the motility and confinement.
Yes. 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 Okay.
I just wanted to.
No. Good. Please ask. Please. So now, so one can also try to look at mechanical properties such as viscosity and means relaxation. Means since these systems are viscoelastic systems, even though I will not be talking about elasticity at all, but we have an intuition that there should be some elasticity which should come into picture, right? So now, what in experiments, what they did, they dissected this PSM even further. They considered three reasons. One is this posterior region of the PSM. This is the middle region of the PSM and anterior region of PSM. So we know that means as all of you have been saying that in the anterior region there will be less motility. Of course, that also implies that the viscosity of the system should be high in that region, right? And similarly, in posterior region, since motility is high, it also reflects that viscosity should be less in that region. And actually, we'll see that that's what happens as well. So, so what they did, they took this, they cut it, and then let it relax. And then what you observe that if there is a fluid kind of system, of course, due to interfacial tension, it will try to capture back its spherical nature, right? So it will try to become spherical in nature. Yeah.
Cells in the walls are not increasing with
Which walls?
like the boundary.
So now we have changed the boundary. So I need to in this. No. So in the explant, there is no addition of the cells at all. Cells are not being added in the system. So there might be some cell division, but that will happen at much later, means longer time scales. Okay, so cell division is not happening. There is no mass injection. You are only looking at the relaxation of the system. Okay, so how the system relaxes. Okay, so you can look at how the rounding happens, and from that one can actually extract a time scale that how fast or slow it is relaxing. So of course, for the posterior region, the relaxation is fast. If there is a relation for the speed with which it will relax, and it is directly related to the viscosity of the system and the interfacial tension of the system. So I'm not going to detail.
On the left-hand side, you are showing the process, or what is right after that?
This one. So these are these parts. You dissect it in 3 parts.
So the cross section.
Yeah, in the cross section. In the cross section, you dissect. So you will have posterior end of the PSM, anterior end of the PSM. Anterior end is not moving. There is not much motility, so it takes really long time to relax and attain a spherical structure. Whereas posterior end is much faster, and that's what they show here also. How the aspect ratio, which is A by B, is changing with time. So for the posterior part, it is really fast, whereas for anterior end, it takes really long time. Okay, one can also measure the viscosity. So they have also measure the interfacial tension for these guys. They have done this micropipette aspiration experiment, which I think Ranjini knows. Okay, so one can measure viscosity, and what you will see that for the anterior region, the viscosity is really large in compared to the posterior end. So we did the same thing in the simulations. We also dissected it in 3 parts. We look at how the relaxation takes place, and also we looked at the viscosity. The inset shows the viscosity. It has the similar trend what you observe here in the experiments also. So basically, if you go from posterior to anterior end, your viscosity increases. Also, corresponding relaxation time to relax to a spherical structure also increases. So this tissue viscosity, which is changing, that largely accounts from this non-uniform mechanical deformation, which you remember the pear-shaped thing. So that can be directly related to, attributed to viscosity. Okay. Now, the one question which I have been posing in the earlier model, that can confinement drive the elongation? To test that, what we did, we took our embryo and we put it in the confinement to mimicking a microchannel setup. And what you do, you just take your, you change your boundary condition in the simulations. You make everything rigid except the tail bud, which still mimics the spring. And then you try to see how the growth of it looks like. So, so basically, in this case, there is no addition of mass, and just the motility gradient, or let's say the high motile cells in the posterior end are able to elongate the PSM. Okay, the same thing. So first we did the experiment, then we requested our experimentalists to do the same thing, and they did. And this is an actual experiment where they take the PSM explant, they put it in the microchannels, and just because of the activity on the PSM is responsible for this elongation. Along with that, we also measured means how important confinements are. So what you can do, you can change the width of this channel, and for the same PSM, you can see how the elongation is going to get affected. So when we change the width, so this black curve corresponding to a width which is 10 cell size, the gray one corresponding to width 20, and the last one is the one which is corresponding to when you kill the motility of the cell. So basically, in the experiments also, you can use the drugs which will kill the motility. In the simulations also, we can do it by hand. We know the parameter exactly. So if you do that, you will see that how the elongation is going to get affected. So you can see that when there is motility and when there is confinement, high confinement, that leads to elongation. And that's what we have been saying from the beginning. The same thing have been observed in experiments also. This is the case when where they are showing that if you change the width of the microchannel from 90 micrometer to 160 micrometer, and the other case where they are comparing it with motile versus non-motile PSM explant. Okay, so one can also do the stress measurement, that how much stress is produced by PSM explant, and we can, so basically this is the experimental setup. You can put a cantilever and from that you can measure the stress.
We
And
are running out of time. Please conclude.
Yes,
We
sure,
have
sure.
another talk from 11:30.
Yeah. I will just, I will end here itself. Okay. So this is the last slide before the summary. Sure. So, so basically, what you can, means what we have shown here, means three different cases. In one case, when you have the PSM explant only in the channel. The second one, when you have PSM, means when you have actual PSM in the embryo. And you can see that in both of these two cases, the stress which is applied by the PSM is different. And the reason being, in one case, you're also adding mass. When you talk about channel, when you talk about PSM explant, there is no addition of mass. Whereas when you talk about embryo, there is a constant influx of mass, constant influx of cells. So that's the reason the stress which is applied by PSM in the embryo is much higher compared to the stress which is applied by a PSM explant. Also, one can do by killing the FGF protein, which is directly linked with the motility. So you can see that if you kill the motility, the stress level goes down. The same thing has been shown in the simulations as well. One can easily control it. And with this, I will summarize this part. So basically, the minimal model which we have shown, it is able to produce the observed effect for PSM explant. And this, these experiments and simulations also confirm that confinement by surrounding tissues in the actual embryo is responsible for this embryo elongation. Okay, so I will stop here. And then if you are interested, then next one means I'll just show the video and I will end here. Sorry. Yeah, so this is how one can extend the model and one can try to show how the somite starts to form. Okay.
Let us thank the speaker.